All right, this is worksheet number 2 of the respiratory system. So let's look at this. Okay, so we're going to be reviewing over breathing mechanisms. So usually you think breathing is just as simple as, it's not. So one respiratory cycle consists of one inspiration followed by one expiration. So you're going to breathe one in, out, that's a respiratory cycle. This is also known as, you can kind of think about this in terms of your tidal volume, which you don't know much about, but we will get through this. The phrenic nerve, so P-H-R-E-N-I-C, phrenic, this stimulates the diaphragm to contract. So your diaphragm is the main muscle of your respiratory system. So when your diaphragm, so we'll get through this. The external intercostals are muscles. Wait, where am I looking at? Okay, so the external intercostals are muscles that kind of line the outside of your ribs. And these contract to expand the ribs during normal, quiet inspiration. So they will expand the ribs. And what's something about really cool about the ribs is your ribs actually move when you breathe, which is pretty wild because we have cartilage in our ribs that allow us to move. So that's interesting. But the external intercostal muscles are the muscles in your chest that do that. But your diaphragm is the main one that controls it. So let's look at number 4. If there is less pressure in the lungs / alveoli, then atmospheric pressure will force air into the airways. So less pressure in the lungs, okay? We're going to think about when you breathe out, there's not too much pressure. Think about your lungs turning to noodles, kind of. Breathe out, kind of feel it. So there's not a lot of pressure. Atmospheric pressure is more outside of, there's more atmospheric pressure outside of the lungs. So pressure and solutes and everything in anatomy moves from a high concentration to a low concentration. There is a high amount of pressure in the atmosphere versus in the lungs. So pressure will force its way into us, because it is high out here and low inside us. You can think about in terms of oxygen. When you breathe out, there's no pressure, there's no oxygen, a lot of oxygen, a lot of pressure out here, so that will force its way into you. Let's see. So then, that's pretty nice, right? Okay, so surface tension aids in lung expansion. This is true. Surface tension is the attraction of water molecules that hold the visceral and parietal membranes together. So surface tension is nice. It's basically the tension on the surface of the lungs, and this kind of allows the lungs to kind of slip and slide and glide within inside you. Versus if there was no surface tension, if there was no, water molecules to help move the lungs and keep them moving, then your lungs really wouldn't move along with your body. This is why if you get like a stab wound, it's pretty bad because it breaks that tension. It interferes with the pressure of your lungs. Quite a few bad things. You don't get stabbed. As a lung expands, compliance increases. This is false. Compliance is known as the distendability of lungs. So this is a property of the lung. When a lung expands, compliance decreases. The lung is not distendable and it is not stretchy. But when a lung shrinks, when you exhale, then what happens to compliance? Well, how distendable is the lung? Compliance is increased and the lung is very distendable. I don't know if that quite makes sense. Basically, is the lung able to expand? If your lungs are already full of air, your lungs cannot expand. But if you don't have any air, then the distendability, the ability for your lungs to expand, the ability for your lungs to distend, distendability, that will be nice. We like that. Okay, so number 7. Elastic recoil is what you put in the blanks, and this occurs during expiration and is when the diaphragm and external intercostals snap back to their relaxed shapes. So when you breathe, it's actually your muscles are contracting to breathe. And then all of a sudden you reach your capacity, okay? More concentration in your lungs, less in the atmosphere. So when your lungs are more concentrated, elastic recoil will be like, and that's when you breathe out. You know? So it's elastic recoil. It's just when it recoils back. It doesn't happen that dramatically, but hopefully that kind of visual helps. And they're now relaxed, which is nice, right? We like breathing out to relax. The next, number 8, resting, normal, quiet breathing is a passive process. This is true. It's very passive. If it were active, we would have to think about breathe in, breathe out, breathe in, breathe out, over and over and over again. You know, kind of like how we have to tell our heart to beat, 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 beat. We don't have to tell our heart to beat. We don't have to tell our lungs to breathe. So therefore, it is a passive process. Although some people can actually control their, that's something else. Okay, resting, normal, quiet breathing, passive process. What muscles are involved in forced expiration? Now we're going to talk about when we forcefully breathe out. This is your internal intercostals and your abdominal muscles. So we're going to squeeze and breathe out. Pull out some yoga mats, maybe, I don't know. So I'm going to pull up a reference sheet really fast that should hopefully help out. So I'm going to go back to this. Wait a minute. So spirometry. I'm pulling out this sheet. Looks like this. It's worksheet number 4, but you don't actually have to do anything to complete it. It's already there. It's just a reference sheet for you to look at. So spirometry, this is known as a spirogram. All right? This top one. Spiro means to breathe. Gram means chart. So that's a spirogram. Spirometry, we use spirograms to measure spirometry. Spiro meaning air and metry meaning measurement. So spirometry is literally the measurement of air. A respiratory volume is a single measurement that can be measured with a spirometer. And a spirometer, if you haven't had lab on already, with Janet or Mark, no, this is Mark's. It's basically you breathe, it's a tube, and you can measure your vital, your inspiratory resolve volume, your total tidal volume, your expiratory resolve volume, and your residual volume. Okay? Your respiratory volumes is a single measurement that can be measured. Your respiratory capacities is a combination of 2 or more respiratory volumes. So I'm going to clip this on and hope this stays. Actually, wait, I can pull it up. So you get this arrow stuff, and you're like, what am I looking at? Because this is kind of scary looking. But this is spirometry. This is a spirogram. But let's look at an alternative, shall we? So I don't know if you can see this. So this is basically your inspiratory reserve volume, your tidal volume, your expiratory reserve volume, and your residual volume. Volumes you can measure within a single setting, okay? Your capacities are all over here. So if you look at your worksheets or you are in an exam, these you have to add up and put into an equation. Which worksheet is this? This is number 2. So as you can see, there are those equations you have to fill out. Sorry for shoving it in your face, but it's right there. So if you look at this, your inspiratory reserve volume, tidal volume, these 3, if you add them up, this will equal your vital capacity, okay? If you look at your, what is this? Your inspiratory capacity equals, this is hard, it equals these two. So you just drag these two across, and it's right there. So these, your inspiratory reserve volume and your tidal volume equals your inspiratory capacity. If you look down here to your functional residual capacity, you will see your tidal volume, expiratory reserve volume, and just drag it across in the chart and it'll equal that. And then over here you have your total lung capacity, which is when you add all your volumes up. So there's that. It's a little brief review, but basically your capacities are made up of volumes and you have to add your volumes. So when you look back up at the chart, basically I took, if you see there's this red arrow here, blue arrow, yellow, orange, inspiratory reserve volume, tidal volume, expiratory reserve, and residual volume. I kind of put those here. 1, 2, 3, 4, 1, 2, 3, 4. All of that there. You just drag them across, and you'll be fine. So that's spirometry. It's kind of how it works out. If there is and the reason why we do this is not to torture you. It's to detect the case of a disease or an illness. So, if you look for cases of, you can look for examples in the disease worksheet that I made. But you can think about, what volumes would be affected by emphysema, how would they be affected? And you won't be asked that in an exam. It's just something to think about. So, I guess moving on from spirometry, we're going to now talk about dead spaces. So your physiologic dead space is basically your atomic dead space plus your alveolar dead space. Your dead space is basically if you breathe in air and it doesn't go to your, so let's do that. This is your bronchial, they're your trachea, your bronchioles, This is in your lungs. Oh, this is nice. So I don't screw this up. Okay, it's a nice lung. Not really. Okay. If you breathe in air, okay, and it gets here, that's nice, right? We want air to get here. But sometimes some air gets into there, and some air kind of just stays in our tubes. It stays in our conduction pathway. And when it stays in our conduction pathway, it's known as dead space. So basically the gist of this is your physiologic dead space is pretty much the same thing as your atomic dead space plus your alveolar dead space, and same thing as your anatomic dead space. I mean, it's pretty much dead space is dead space. So moving on to some calculations, and you can do these. You have your minute respiratory volume and your alveolar ventilatory, oh shoot, I forgot what that stands for. Alveolar ventilation respiration, I think. MRV is the volume of new air that moves into the respiratory passage each minute. So MRV is calculated by your respiratory rate times your tidal volume. Your respiratory rate is how many times you breathe. The regular person breathes 10 breaths per minute. So MRVs is respiratory rate times tidal volume. So while respiratory rate is how many times you breathe per minute, your tidal volume is how much you breathe. What is the volume of how much you breathe every minute? So that's minute respiratory volume. And, there's an equation. And you may have equations on your exam. The next one is trying to calculate the AVR. And this is calculated by taking your tidal volume minus the physiologic dead space. You're going to subtract those two first. And then you're going to multiply that value by your respiratory rate. So the reason why is AVR is asking for how much oxygen gets into your lungs. How much do you actually breathe? Because you can breathe a lot, but if it doesn't get into your lungs, then what's the point, right? So that's what AVR measures. So tidal volume being the amount you breathe, and you're going to take that amount you breathe, so you're going to take your tidal volume, and you're going to subtract it from all this dead space. Okay? You don't care about this stuff, so you're going to subtract the physiologic dead space from your equation, and you're just going to be wanting to focus on this. You have to subtract the two to get that. So your tidal volume minus your physiologic dead space. This is a minus sign, I guess. And then you're going to multiply that by your respiratory rate, which is how many times you breathe per minute. And that's how you get your AVR. Let's see now. Number 15, what are some non-respiratory movements? Sneezing, crying. Those are non-respiratory. You don't really breathe. Hopefully you don't do that prolonged. So what are some respiratory areas that control breathing? You have your medullary respiratory center and your pontine, I like that word, pontine respiratory group. Your medullary respiratory center is, and these are both in the basal part of your spine. And this connects your brain to your spinal cord. So in your medulla, the ventral part, which means the front part of your medulla, is responsible for the rhythm of breathing. So it sets the rhythm. It's like, yeah, breathe slow, breathe fast. Your dorsal respiratory group, dorsal meaning behind. So this is so when you look at your medulla, if you were to split it down the middle and look at it, your medulla is up here. The ventral side would be the respiratory rhythm. The dorsal side would be back here. And the dorsal side is responsible to stimulate muscles for inspiration. So this is important because if you get this damaged, you damage your dorsal part, you're not going to breathe. So it's really important. Next, you have your pontine respiratory group. And this has to do with your pons, because you have your pons in your medulla, which is right next to each other, but your pontine portion limits the duration of each inspiration and helps regulate respiratory rate. So your pontine is kind of like I said earlier, if you have a bunch of carbon dioxide inside of you, that's a waste product, you're going to want to get that out. So to get that out, you're going to breathe really, really, really fast. You're going to have hyperventilation. But if you don't have enough carbon dioxide, your body's going to want to retain it, so you're going to breathe a little bit slower and hypoventilate. That's what your pontine does, respiratory group. Number 17, which among these factors, which all affect rate and depth of breathing, is the main controlling factor? And although all these factors are important, the one that really regulates the rate and depth of breathing, like I said earlier, is your carbon dioxide. Literally, you breathe based on how much waste you have in your body. Another thing that affects this is carbon dioxide is an acidic substance, okay? Our bodies are naturally alkaline, so having an acid in our body throws off homeostasis. It throws it out of, throws us through a loop. So carbon dioxide causes us, it causes our pH to turn acidic, and when we're acidic, we don't like that. So we breathe faster, and that controls the rate and depth of breathing. This next question, 18, low blood oxygen. So this is the statement. Low blood oxygen, so PO2, I usually think about in terms of pressure of oxygen. Low blood, PO2, has a huge impact on central chemoreceptors. This is false. This is because, the true answer to this would be carbon dioxide, and there's a rationale in there. Yeah, and you'll learn about this too as we move on to worksheet number 3, which has to do with gas exchange. That one's fun. So going on, number 19, central chemoreceptors monitor pH in the brain. Okay, central chemoreceptors. Central, meaning in the center. Peripheral chemoreceptors monitor pH in the carotid and aortic bodies. And carotid and aortic bodies, I should have been more descriptive, but these are arteries. Your peripheral chemoreceptors are just looking for pH in your arteries. So while you have some in your brain, because your brain is your main priority, you have central chemoreceptors there to protect the brain, but then you also have peripheral ones in your arteries, so you're still able to be regulated. Yeah, so 21, low blood PO2 has a huge impact on central chemoreceptors is true. So this top one was talking about low blood PO2. Wait a minute. That first one is wrong. Okay, number 18 is wrong. You might as well just forget about that, but keep that rational. I think actually, in number 18, made a typo. Replace PO2 with H plus. Yeah. Okay. So low blood PO2 has a huge impact on central chemoreceptors. This is true. A significant decrease in PO2, which is the pressure of oxygen, so a significant decrease in oxygen causes an increase in breathing rate and tidal volume. So if you don't have oxygen in your brain, you're going to breathe a lot faster. And this will increase alveolar ventilation, which alveolar ventilation is how well your alveoli breathe. This is not caused by a major response until, this doesn't actually, your PO2 receptors don't actually kick in until you lose about 50% of oxygen. And by that term, you're not in a good place. So moving on, Herring-Brewer reflex prevents overinflation of the lungs. So when you breathe out, so a reflex is something, just say you hit someone's knee, okay, and your knee flies up. That's a reflex. It's something that happens automatically in response to something that's kind of irritating. So when you breathe in, your Herring-Brewer reflex will be like, wait, whoa, what? No. And then you're going to breathe back out. So, and it is stimulated by stretch receptors because our lungs are stretching. The sensory impulse travels up your vagus nerve and into respiratory areas. The deflation reflex promotes lung collapse is false. It is true. The deflation reflex will prevent your lungs from collapsing. So it allows your lungs to push out. So you have reflexes. You breathe out. Your Herring-Brewer's like, what are you doing? You breathe that out. Your deflation reflex kind of is like, what are you doing now? Breathe in. I am shriveled. I don't know. So let's keep going. Mechanisms that increase breathing rate is your cerebral cortex in the joint reflex. This has to do with receptor cells, muscle, and proprioceptors that are stimulated by muscle movement. These are mechanisms that increase breathing rate. The site of gas exchange in the lungs. The site of gas exchange is the lungs. That statement is false. The site of gas exchange, again, is your alveoli, which are kind of like the fruit of your bronchial tree. And this next question is, again, about alveoli. So what are 3 cell types of alveoli? Well, you have simple squamous, type 1, that make up most of alveoli. And simple squamous tissue is very, very thin. And you want this very thin so oxygen can come through and carbon dioxide can leave. You don't want something thick because then oxygen and everything is just going to bounce off and it won't work out. So you have simple squamous, which is great. And this makes up most of the alveoli in the lungs. You then have alveoli and macrophages. And macro means big and phage means eat. And I think about these guys in terms of like Pac-Man in your lungs that kind of eat debris or eat just, the little things like you don't need. You could live without. Literally, you could live without. Or you will live without. Okay. The next type of alveoli you have is septal cells. These are type 2 alveoli, and these secrete surfactant and reduces surface tension. So surface tension, this is your lung, right? Surface tension is great outside of your lungs, okay? Because it allows your lungs to be attached to your pleural membrane. So surface tension is great out here. But when it's in your alveoli, not so much. You don't want surface tension. So type 2 alveoli decrease surface tension within our lungs, which is really nice. Let's see. Number 27, gas exchange occurs between alveolar air and blood through respiratory membrane. This is true. Gas exchange does occur between the alveolar air and the respiratory membrane. So that's true. 3 structures of the respiratory membrane are the alveolar wall, the blood capillary, and the basement membrane. And we will go more into this. Actually, let's pull up a picture. Respiratory membrane. If you want to see this, that's nice. So as you can see, this pinkish portion of it is your alveoli, and this is your capillary. And the basement membrane is this this area kind of between, so to speak. But yeah, super important. Respiratory membranes help us breathe, and we like breathing, right? So molecules move from a region of, like I said earlier, okay? In general, molecules move from a region of high concentration toward another region of low concentration, always. So with gases, gas diffuses from an area of high parietal pressure. So pressure, just think about in terms of concentration, if you have had chemistry, high parietal pressure to low parietal pressure. And parietal pressure is just fancy for pressure. So it goes from high concentration to low concentration. So if I breathe in, my lungs are like balloons, okay? They are more able to pop, and that's because they have a lot of pressure in them, okay? So if there's high pressure in my lungs, high pressure in my lungs and lower pressure out here, then I'm going to breathe out naturally. But if I don't have any pressure in here, my lungs are like limp, okay? That's a low concentration of pressure, while there's higher concentration out here. So it's going to force air into me and forces me to breathe. I don't know how much of a visual too. So looking at the rationale, factors that increase diffusion across the respiratory membrane, more surface area, shorter distance, greater gas solubility, and a steeper parietal pressure gradient. Then there are factors that decrease diffusion across the respiratory membrane, and these are a decrease in surface area and harm to the respiratory membrane. We want factors that increase diffusion. We want more surface area, shorter distance, greater gas solubility. And solubility means dissolution. Does gas dissolve? And by that, it's talking about, does gas dissolve from the respiratory alveolar membrane into the blood or not? We want more of that. If our alveoli are damaged, though, so just say, we have emphysema, which is when your alveoli, so if you think about, so if you think about your lungs in terms of a tree, and if that fruit, the alveoli, if they rot, think that's what emphysema does to your lungs, basically. So if it causes that rotting to happen, you're not going to have any gas exchange, and therefore the gas solubility is going to be low. Another thing that we do want is a steeper parietal pressure gradient. And by that I mean if the gradient, so a gradient is like a scale. And we want that scale to be very unbalanced back and forth, because the more unbalanced it is, the more likely we're going to be able to breathe in and out easier. The issue is if that isn't stable. If it is stable. So if we have, a pressure of 20 in our lungs and a pressure of 20 in the atmosphere, we're not going to be breathing in and out. We're just not going to breathe. Unless we do it forcefully, of course, we will survive. But it needs to be more, we need to have it changing within our lungs and within our atmosphere in order to keep it a quiet, normal, passive respiration. There needs to be a difference, a steeper gradient, a difference in concentration. So the next portion of this, number 30, driving force for diffusion of oxygen and carbon dioxide across the respiratory membrane, is a difference in parietal pressures is true. Diffusion of oxygen and carbon dioxide depend on the difference in parietal pressure. We need to have a difference. Those scales need to be constantly unbalanced, okay? So going through this, this just asks you, up here, you're just going to label each one. You're going to come up with a definition on your own. And actually, no, this is, yeah. And then you're going to put the equations on this side. Other than that, that concludes worksheet number 2. And it's pretty much tied. So, that is that.